You know how sometimes you learn a rule and it seems totally solid until it just isn't? Well, in science, those little cracks in the rules are often where the most amazing discoveries happen. Today, we are diving headfirst into a fantastic genetic puzzle that for a while seemed to completely break the fundamental laws of inheritance laid out by Gregor Mendel. It's a real story of mystery, of some seriously clever scientific detective work, and a discovery that forever changed how we look at our own DNA.
All right, so here's our game plan for cracking this case. First, we're going to look at the totally baffling pattern that first told scientists, hey, something weird is going on here. Then, we'll investigate two key sets of clues, the very strange case of coupled genes and the equally bizarre case of repulsed genes. After that, we'll see how the code was finally cracked, which leads us to the big reveal, the truth hidden right inside our very own chromosomes. So, let's get into it. So, our story kicks off with a pretty major scientific mystery. Back in the early 20th century, Mendel's laws were, well, they were everything, the absolute foundation of genetics. And one of his big ideas was the law of independent assortment. Think of it like this. It basically says that the gene you inherit for, say, eye color has absolutely nothing to do with the gene you inherit for whether you have a widow's peak. They're supposed to sort themselves out independently, kind of like shuffling two completely separate decks of cards. But two scientists, William Bateson and Reginald Punnett, they found something in their experiments that just didn't add up. And that was the big question staring them right in the face. They were studying two traits in sweet pea plants, flower color and the shape of the pollen. Now, according to Mendel, getting purple versus red flowers should have been a totally separate event from getting long versus round pollen. But the numbers they were getting in the offspring, they just weren't lining up with his predictions. It was almost like the gene for flower color and the gene for pollen shape were somehow stuck together, refusing to go their separate ways. This observation, as simple as it sounds, was the first major crack in a very established scientific law.
So, what should they have seen? Well, based on Mendel's law, when you do what's called a dihybrid test cross, which is just a fancy way of saying you're testing two traits at once to see what's going on, you expect a really specific, really clean outcome. You should get 4 different genetic combinations in the kids, and the key thing is they should show up in roughly equal numbers. As you can see here, you're supposed to get this beautiful 1-to-1 to 1-to-1 ratio. Each possible combination should make up about 25% of the total. That was the textbook answer. That's what should have happened. But look at this. This is what they actually found. And the results weren't just a little bit off. They were wildly, dramatically different from that 25% expectation. I mean, instead of 4 equal piles, they got 2 massive groups, each making up almost 44% of the offspring, and then 2 tiny little groups at just over 6%. It's not even in the same ballpark as a 1-to-1 to 1-to-1 ratio. Clearly, something else was going on here. Something powerful enough to totally ignore a fundamental law of genetics. Okay, so let's put on our detective hats and really dig into this first cross. What could possibly explain these wacky numbers? Well, the scientists gave this phenomenon a name. They called it coupling. It was as if the dominant traits were physically stuck or coupled to each other, and the recessive traits were too. This was the first huge clue in solving our genetic mystery. Okay, so how do they actually get these results? Let's walk through the setup. First, they started with purebred parents, meaning one parent was dominant for both traits. We'll show that with capital letters, BBLL. Dominant just means if you have that version of the gene, you'll see that trait. They crossed that plant with another one that was recessive for both traits, little letters, BBLL. You only see those traits if there's no dominant version around. Now, the first generation of offspring, the F1 generation, were all genetically identical, BBLL. They had one of each version. Then came the really important step, the test cross. They took this F1 plant and crossed it with a fully recessive one. And this is a super clever trick geneticists use because the recessive parent can't hide anything. It lets you see exactly which gene combinations the F1 parent is passing down. And the result of that cross was that totally skewed data we just saw. Now, to make any sense of this, we need to sort the offspring into 2 really important groups. First, you've got the parental types. These are the offspring that have the exact same combination of gene versions or alleles as the original grandparent generation. So in this experiment, that's the dominant-dominant combo and the recessive-recessive combo. Then you've got the recombinant types. Recombinant just means recombined. These are the kids with the new, mixed and matched combinations that you didn't see in those original grandparents. And trust me, this difference between parental and recombinant is the absolute key to unlocking this whole thing. And this chart just makes it impossible to ignore. I mean, look at that.
Remember, we were expecting something like 50% parental types and 50% recombinant types, but that is not at all what happened. When you add up the numbers, the parental types made up a whopping 87.4% of all the offspring, and the new recombinant types, a tiny little 12.6%. The evidence was just overwhelming. The original combinations of alleles were being passed down together way, way more often than they should have been. It's almost as if the B and L alleles from one grandparent and the B and L alleles from the other were physically chained together and refused to let go. But just when they thought they were getting a handle on it with this coupling idea, the plot thickened. You know, like any good scientist, Bateson and Punnett ran another experiment to check their work. But this time they switched up the starting parents. And what they found was just as weird, but in a completely opposite way. They called this new phenomenon repulsion.
Okay, you really got to pay attention to this setup because this is where it gets super interesting. This time, they started with different purebred parents. One had a dominant trait and a recessive trait, BB small l, and the other had the opposite mix, BB capital L. Now here's the really crucial part. When they crossed these two, the F1 generation that they got was still genetically identical to the F1 from the first experiment. Every single one was BBL. So they took this identical plant and did the exact same test cross. Now, logic would tell you that if the plant is the same, the results should be the same, right?
Wrong. So wrong. The results were completely flipped on their head. Now, the BBL and BBL types, the ones that were super common before, are the rare ones at only 6.3% each. And the BBL and BBL types, the rare ones from the first cross, are now the overwhelmingly common ones at almost 44% each. The numbers are basically identical, but the common and rare groups have completely swapped places. I mean, what on earth is going on here? So, to make sense of this flipped result, we have to relabel our categories based on the new starting parents from this second cross. Remember, this time the grandparents were big B little l and little b little l. So now, the parental types are the offspring that have those combinations, the big B little l and little b little l ones. And the recombinant types, the new mixes, are the big B big L and little b little l ones. The big insight here is that these labels aren't permanent. It all depends on how you started the experiment. And when we recategorize the results this way, bam, the hidden pattern just snaps right into focus. Look at that. Once again, the parental types make up 87.4% of the offspring, and the recombinant types are just 12.6%. The specific gene combinations flipped, but the ratio is identical to the first cross. This proved there was a deep, underlying rule at play. Whatever combination of alleles you start with in the grandparents is overwhelmingly likely to be passed on together. This wasn't some kind of magic, it had to be something physical. So now we have our two key pieces of evidence. We've got two experiments with what look like opposite results, but they're both being driven by the exact same hidden pattern. This led the scientists to give these two conditions formal names, which really helps us wrap our heads around the mechanism behind it all. So that first experiment is what they called the coupling state, or sometimes the cis configuration. Think of it like this. The dominant alleles, B and L, came in on the same team from one parent, while the recessive alleles, B and L, came in from the other parent. They're inherited as a block, they're coupled. The second experiment shows the repulsion state, or transconfiguration. Here, each parent gave a mix. One gave a dominant and a recessive allele, B and L, and the other gave the opposite, B and L. It's almost like the dominant alleles started out on different teams, and they wanted to stay that way. They repel each other. What's pretty amazing is that Bateson and Punnett saw all of this, they documented it, but they couldn't fully explain the physical reason why it was happening. They knew genes were part of the story, but the actual mechanics were still a black box. The complete explanation had to wait for something called the chromosomal theory of inheritance to be fully fleshed out. And as this quote tells us, the answer wasn't some mysterious force messing with the genes. It was in the physical reality of the chromosomes themselves. And this is the final piece of the puzzle. We're about to see how the physical arrangement of genes on chromosomes perfectly, beautifully explains everything we've seen so far. This is the big aha moment. This slide right here reveals the hidden truth. While those two F1 plants were genetically identical on paper, they were both BBLL, their chromosomes told a very different story. In the coupling state from cross 1, one chromosome physically carried both dominant alleles B and L, while its partner chromosome carried both recessive alleles B and L.
But in the repulsion state from cross 2, each chromosome carried a mix. One had B and L, and its partner had B and L. This physical arrangement, this linkage on the chromosome, that is the key that unlocks the entire mystery.
So how does this chromosome setup lead to those skewed numbers? It all comes down to a process called meiosis, which is the special cell division that creates sex cells, like pollen and ovules. In the coupling state, when the plant makes these cells, the default outcome is that the chromosomes just stay intact. This produces gametes that are either BL or BL, our parental types, and that's why they are so common. The only way to possibly create the new combinations, BL or BL, is that the two partner chromosomes actually physically swap pieces. This event is called crossing over. You can imagine them getting tangled up and exchanging a little bit of information. But since this physical swap doesn't happen every single time, those recombinant gametes are much, much rarer. And this perfectly explains why the parental types were so common in cross 1.
And this is exactly why the second cross was a perfect mirror image. In the repulsion state, the starting chromosomes were BL and BL. So during meiosis, the common, default, non-recombinant gametes are BL and BL, which you'll remember were the parental types for that cross. To get the other combinations, you again need that relatively rare crossing over event to happen, so they become the rared recombinant gametes. It's just incredible. The exact same physical mechanism, the occasional swapping of chromosome parts, perfectly explains both the coupling and the repulsion results.
So if we boil this all down, the ultimate takeaway is this. Genes that are located on the same chromosome are physically linked together, kind of like beads on a string, and they tend to get inherited as a single package. This is why Mendel's law of independent assortment breaks down. It really only works for genes that are on different chromosomes. The only thing that can break this physical linkage is crossing over. But because crossing over is a game of chance and it doesn't always happen between two specific genes, the original parental combinations are always going to be more common than the new recombinant ones.
And this leaves us with the most powerful idea of all. Think about it. If the percentage of recombined kids is really low, it must mean that crossing over rarely happens between those two genes, and that probably means they're super close together on the chromosome. If the percentage is higher, they must be farther apart. Just by counting the different types of offspring, geneticists suddenly realized they could create a map showing the exact order and relative distance between genes on a chromosome. This perplexing puzzle, this violation of a rule, didn't just solve a mystery. It gave us one of the most fundamental tools in all of genetics, and it paved the way for everything from understanding genetic diseases to mapping the entire human genome.